Hello, everyone. This is a good morning to you all from here in US. And today, this is the inaugural lecture, which is organized by Pathcast and in collaboration with Association of Indian Pathologists of North America. And this inaugural lecture will be delivered by none other than Professor Vinay Kumar, who is from University of Chicago, and those who have read Robbins Pathology. So we don't need to introduce him again, and everybody knows him. So we are honored to have Professor Kumar addressing this inaugural lecture for IPNA. And this lecture is titled Molecular Biology of Cancer and Paradigm Shift in Cancer Care. Without much ado, over to Professor Kumar now. And thank you for joining us, Professor Kumar.
Thank you very much, Rifat and Emilio. I'm delighted to be here. I hope all of you can hear me. And it's good morning in Chicago, but it might be good evening, good night, depending on where you are. It is Namaskar, Jai Hind, Salam Alaikum, Sat Sri Akal, whatever appeals to you. And with that, we will start this. Just wanted to talk a couple of minutes about the overview of what I'm going to present. What I'm going to present is this, the molecular basis of cancer, okay? And just the general principles using some examples. It is not going to be exhaustive, okay? It is not going to be very detailed, but I will go into details enough so that I can point out to you how cancer therapeutics can be developed and are being developed by understanding the signals, the molecular signals, and molecular basis of cancer. Again, it's not going to be in detail, in depth of any specific aspect. My goal is to go overall, but the goal is, of course, for you to see how therapies are developed or can be developed by an understanding of the molecular biology of cancer. That's the goal of this lecture, and I think it is stated in the next slide. To discuss selected genetic changes that are responsible for hallmarks of cancer. We'll come back in a minute. To provide examples of how cancer treatment can be directed to cancer-causing mutations. To discuss why the currently available treatment is inadequate. And to describe how the genomic profiling of the cancer may make the treatment specific for the mutations that drive the cancer in a given individual. That is really the holy grail of personalized medicine, that everybody gets a specific treatment that is specific for the individual and specific for their tumor, rather than how we do it currently. We give everybody the same chemotherapy. And you hear the data, like, for example, it say, this particular chemotherapy protocol had a 60% response rate, remission rate. What does it mean? 40% got the medicine, got all the side effects, but did them no benefit. And so the goal of personalized medicine is that no one should get the therapy that is not appropriate for them. Okay. We'll go over some of these fundamentals, which will be illustrated more nicely in the next slide. So I think many of these things you all learned in your medical school classes, and so none of these, what I'm writing here, is showing you here, is novel or unique, but it's good to have an overall view of this, and a couple of important points in there. One is that cancer is a genetic disorder caused by mutations in DNA. These mutations are usually acquired in the somatic cells, but in some cases are inherited in the germline, and that's when we see familial cancers, like, for example, retinoblastoma or Wilms tumor or melanoma, and so on. It's important to note that no single mutation is sufficient to transform the cell. No single mutation can transform the cell. Carcinogenesis actually is a multi-step process resulting from accumulation of multiple genetic alterations. So I think it's important to note that it's not just one target, one cancer, one target. The initial target of mutations is in a stem cell with self-renewal capacity. Obviously, for cancers to grow, the cells must keep on dividing and self-renew. Now, a normal cell can renew themselves up to a certain point and then they die. Cancer cells have figured out a way to keep on dividing, as many of you know, by activating the telomerase. The initiating mutations in the stem cells are heritable and are passed to all the progeny cells. Hence, all cancers are clonal. All cancers are clonal. Every cell within a cancer that is clinically visible has been derived from a single cell. The clonal progeny of the transformed cells accumulate additional mutations that occur randomly and are subjected to Darwinian selection, survival of the fittest. And we'll come to that in a minute. As a result of continuing mutations and Darwinian selection, even though malignant tumors are clonal in origin, they are heterogeneous by the time of clinical presentation. And this is also an important concept. And finally, mutations impart to cancer cells a set of properties called the hallmarks of cancer. And we will discuss, the whole lecture will be around hallmarks of cancer. Okay, let's take a look at the next slide. This shows graphically that cancer arises from a mutation in it's a normal cell. It's a normal stem cell. Okay, and there's an initiating mutation which is not sufficient to cause transformation. And then with passage of time, additional driver mutations occur. And during this time, the cancer acquires the hallmarks of cancer, which will come in a minute, include invasiveness and uncontrolled growth or resistance to apoptosis and so on. Okay. And there is further genetic evolution and more mutations occur, and these mutations occur randomly in these cells. In other words, all the cells are the progeny of a single transformed cell, but once they start growing and accumulating more mutations, those mutations don't occur in every cell. Some cells will get mutation X, some will get mutation Y on top of the original mutation. Okay. Now, the importance of this is that some of these mutations will make the cells aggressive. Okay, they'll give them the property of growing with very small amount of growth factor. Okay. Some of them will give them the property of evading immune responses. Some of them will give them the property of activating the telomerase. Okay, so in the cancer that one sees, okay, like this, you see the multiple cells with different colors. That's to indicate that each one of these are subclones of the original clone, and they undergo Darwinian selection. So if a mutation occurs in one of the cells, which makes it a target for T lymphocytes, it will die. So just like Darwin's hypothesis had survival of the fittest, Darwin's hypothesis works here also. There is survival of the fittest. That is, Darwinian selection ensures survival of cells that beat the odds. That means, that escape this, that escape that, they can divide, they can move, they can inactivate lymphocytes and so on and so forth. Okay, so just quickly to go over it again, cancer arises when mutations in stem cells, because by definition they can renew themselves. No single mutation can cause cancer, as I showed you here. Some mutations affect genes that regulate cell growth, and these are called driver mutations. And other affect genes that are not involved growth control are called passive mutations. Okay, they're just for the ride. Mutations are random, and they do not affect all cells equally, producing a clonal but genetically heterogeneous cancer. Darwinian selection ensures that survival of the cells that beat the odds. So by the time human tumor is visible, okay, it has already undergone all these steps, and sometimes these steps could take years, and before sort of the cancer is manifest. So, but by the time the cancer is manifest, multiple mutations have occurred. Darwinian selection has occurred. So it's a clonal but heterogeneous population with each group of cells having slightly different properties. So as mentioned earlier, mutations impart to cancer cells a set of properties that are called hallmarks of cancer. Okay, we shall go further. So here is a sort of wheel-like diagram which you might have seen earlier in various books and textbooks. It is from Hanahan and Weinberg's classic paper in Cell. And it lists 8 hallmarks of cancer and 2 enabling factors. Okay, so the hallmarks of cancer are sustaining proliferative signaling, that is, keep on growing even in the absence of growth signals, not responding to feedback mechanisms, not responding, evading growth suppressors, not responding to physiological signals that suppress growth, avoiding immune destruction. Okay. Then enabling replicative immortality. That is, keep on dividing without undergoing exhaustion, so to say. Activation of metastases and invasion genes, okay? Inducing angiogenesis. Cancers can't grow beyond a couple of centimeters without blood vessels forming. So to be able to form blood vessels is a hallmark of cancer. To be able to resist death is a hallmark of cancer. Okay, to have a dysregulated cellular energetics, one of the novel hallmarks that has been discovered the last several years. We'll talk about it. Okay, and there are two enabling properties: tumor promoting inflammation, okay, and genomic instability. So what I will do is this: I will go over some of these. Okay, and as I go over this, I would like to sort of be able to explain to you how mutations change the properties of cancer, because the treatment of cancer will eventually have to be to overcome the effects of various mutations. Okay, so this is going to be a series of diagrams in which I will go over with you how does a signal that comes out of growth signal that comes from outside the cancer cells, the growth factors, how does it go through the cell membrane and across the cytoplasm to the nucleus, where the genes that are required for cell division have to be activated. Okay, so in a resting cell, normal physiological cells, everything is quiescent. Okay, it begins it begins by a growth factor binding to its receptors. Okay, very simple to understand. And what I'm showing you is a tyrosine kinase type receptors. There are other receptors as well. Okay, the growth factor binds to its receptor. Okay, then what happens? There's a transient and limited activation of growth factor receptor. So there's an activation of this receptor, and the activation of these receptors recruits signal transducing proteins on the inner leaf left of the plasma membrane. In this case, the signal transducing protein, which is the very common signal transducing protein that I've shown, is the RAS protein. It's product of the RAS gene. So normally RAS is sitting in an inactive form with a guanosine diphosphate attached to it. Okay, with the activation of the growth factor, a signal is sent to the inactive RAS, and it adds one more phosphate and changes from GDP RAS to GTP RAS. Okay, one can see that here. It's activation of this. But normally the GTP RAS, which is active RAS, is very quickly returned back to GDP RAS, which is inactive, by hydrolysis of GTP. Now imagine a mutation in active RAS, which abrogates this property of hydrolysis of GTP. RAS is stuck in activated state. Okay, so what does it mean? The RAS alone can't do anything. RAS has now to talk to the nucleus, right? Okay, so here we show RAS talks to the nucleus. So what's happened here? Transmission of transduced signal to the nucleus by a cascade of signaling transducing molecules. So active RAS, during the time it's active, unleashes, opens up many different pathways of signal transduction, which eventually impact on the nucleus and activate transcription of genes that are required for cell division. These include proteins like PI3 kinase, ACT, mTOR, RAF, MAP kinase. There's no need for you to memorize these things, but except to understand that to go for the signal which started with the growth factor and receptor, went through RAS, has to be sent to the nucleus, and there are many steps in between that go to the nucleus. Now imagine, what I told you earlier, the mutation in RAS will trap RAS in the active state. By the same token, mutation in PI3 kinase, mutation in RAF, mutation in ACT, anything that's along this pathway, okay, if it mutates, okay, it could lead to a state of sustained signaling, and the cell thinks it has to divide, even though there is no growth factor stimulus coming from there. Okay, so the transcriptional activation of genes required for cell division, entry of cells to the cell cycle. Okay, having said that, I think it is easy for you to understand that mutations in this pathway that we just described, starting from the growth factor receptor to the transcription factors to these cell cycling proteins, in this pathway, mutations pathway cause sustained and uncontrolled growth by conversion of normal growth regulating genes, which are called proto-oncogenes, to oncogenes. So this is not rocket science. Proto-oncogenes are these genes that encode for RAS, genes that encode for growth factor receptor. When they are normal and they're functioning normally, we call them proto-oncogenes because they are precursors of oncogenes. Mutations convert them into oncogenes, where from a regulated state, they go to an unregulated state. Okay, so let's first see some examples on mutations or changes that affect growth factor receptors and how they are involved in various forms of cancer. Okay, so growth factor receptors and cancer. So growth factor receptors can be modified or mutated in two fundamental ways. One is by amplification or expression. So if the growth factor receptors amplify and the density of the receptor on the cell is, let's say, 10 times that of a normal cell, what will happen? Even with very small growth signals, very tiny amounts of growth factor, okay, the pathway will be activated. Pathways which we showed started from here. Now, if there's an amplification of growth factor receptor and there are full of receptors like this, then even small amounts of growth factors will trigger the whole process, okay, and confer upon cells the property of division. So, for example, EGFR, a member of the EGFR family of growth factor receptors, ERBB1, is overexpressed in squamous cell carcinoma of the lung, head, neck, and glioblastoma. 80% of them have it. Glioblastoma, about 50% have it. Similarly, a very well-known growth factor receptor called HER2/neu or ERB2 is expressed or amplified in 25% of breast cancers, some adenocarcinoma of the lung, ovary, salivary gland. This is just, you see, what this means is this: changes in this receptor are fairly common in different form of cancer. The other way growth factor receptors can dysregulate growth is by mutational activation. Okay, that was overexpression. This is activation. Now, what activation does is it sends growth signals without any growth factors. Here, they could respond to small amount of growth factors. Here, the mutations make the receptor believe that a growth factor is constantly stimulating it. And so these mutations convert proto-oncogenes into oncogenes. Okay. For example, FLT3 in chronic myeloid leukemia, a KIT in gastrointestinal stromal tumors, GIST, platelet-derived growth factor receptor, also in GIST, epidermal growth factor receptor in adenocarcinoma of the lung. So what's the importance of all of these things in terms of personalized therapy and therapeutic implications? We know that those that express those that express the HER2/neu, they can be targeted by a drug called Herceptin, which actually is very beneficial for the cancer patient. So right now you see we are stratifying cancer patients. We are stratifying breast cancer patients into those who have this abnormality, and if they do, the treatment for them is this. Already we are going towards somewhat of personalized medicine, not personalized to the level of individual, but personalized to the level of a group of people who share a common genetic abnormality. Similarly, the imatinib or Gleevec is used for GIST, okay, and not for every type of GI cancer, only those that have the KIT mutation. And similarly, those lung cancers which have EGFR receptor mutations respond to Tarceva. So this is the sort of the beginning of stratification and beginning of going towards personalized medicine. Why? Because we understand the molecular changes and the drugs have been developed to target those molecular changes. So as I mentioned, there can be mutations anywhere in this pathway. We just gave an example of this one. We'll give now consider the example of mutations in the RAS gene, which are very common and occur in 30% of human cancers. Of all the oncogenes, RAS is the one that is the most commonly mutated in human cancers. It is not the most commonly mutated gene in all cancers, because the most common gene mutated in all cancers is P53. This, amongst oncogenes, RAS is the most common. Okay, so So mutations in signal transduction pathways originating from receptor tyrosine kinases. Okay? And so RAS, if there are many cancers, 30% of human cancers have point mutations that trap RAS in GTP1 activate stage. Remember, I told you earlier that a mutation in active RAS can prevent it from going to the inactive state, and because the GTPase activity is no longer there or markedly reduced. Okay, so RAS is now stuck in active state. If it's stuck in active state, it keeps on signaling, and the cell keeps on dividing. Similarly, it's interesting for you to note that RAS can be activated either by mutation in RAS itself or mutations in GTPase activating proteins such as NF1, the gene that is affected in neurofibromatosis. It also functions by just changing the properties of RAS protein such that it is stuck in GTP bound state. There are mutations downstream of RAS. We saw that there are many other things that occur downstream of RAS. Okay? So any mutations in these pathways starting from here will mimic each other. And downstream of RAS are important mutations. For example, BRAF mutations in many cancers, particularly melanomas, okay? PI3 kinase in many cancers. And again, coming to personalized medicine and therapeutic implication, BRAF mutations, there's a drug for BRAF mutations. And so let me just first sort of give you a slide to give you an idea how personalized medicine may work. So we talked about paradigm shift. Okay? We classify tumors on the basis of the site where they are, their morphology, whether they are squamous cells or adenocarcinomas or small cell carcinomas or large cell carcinomas. But guess what? These 4 tumors, melanomas, most of them, a proportion of colon adenocarcinomas, Langerhans cell histiocytosis, all of hairy cell leukemias, a fraction of papillary thyroid carcinomas share one thing in common. They all have exactly the same BRAF mutation called V600. Okay, so the provocative thought is these histologically diverse tumors have the same mutations. Should we call them BRAFomas? Because after all, the driver mutation is BRAF. That's what matters. That's what counts. I don't think we shall do that, okay? Because there are subtleties in this. Whereas melanoma responds exceedingly well to drugs that inhibit the BRAF mutation. Colon adenocarcinoma does not. There are recent papers which show that for these inhibitors to work in colon carcinoma, there have to be antagonists of the EGFR as well. So colon carcinoma, unlike melanoma, has a sort of multiple driver pathways. One driven by BRAF mutation and one driven by EGFR mutation. And if you cancel one of these two, the tumor still grows. This is again an example of how our treatment is getting more sophisticated. And so for example, only those adenocarcinomas that have the BRAF mutation, it's about 20% or so, I think, okay, are going to be treated like this. So it's important to have a molecular diagnostic test which can tell us which cells, which tumors have these kinds of mutations. So we talked about receptor kinases so far. There are also non-receptor kinases in the cell, and there are 2 examples of that which have become therapeutic targets. One is the BCR-ABL translocation that occurs in chronic myeloid leukemia, classical 922 translocation. It forms the BCR-ABL fusion protein, which has sustained tyrosine kinase activity. It drives central CML. It's interesting. There's a phenomenon called oncogene addiction. What does it mean? It means that even though a full-blown CML has more than BCR-ABL translocation, so that's a driver mutation, but this is not the only mutation in the cell. Okay, as we mentioned earlier, tumors arise by accumulation of multiple mutations. But in this case, what has happened is this: that we can treat it with a drug that antagonizes BCR-ABL tyrosine kinase activity. And you might say, well, what about all the other genes that accumulated in CML? True, but it seems the function of all those other genes is also dependent upon there being a BCR-able fusion protein. So this is called oncogene addiction. So CML cells are addicted to BCR-able, and for us it's a good thing because we can target it. JAK2 mutations result in sustained activation of the JAK-STAT signaling pathway, and this occurs in chronic myeloproliferative disorders such as polycythemia vera, primary fibrosis, and there are antagonists for JAK2 mutations. Now, notice this. If you look at all of these diseases, CML, polycythemia vera, primary fibrosis, they're all myeloproliferative disorders. They're all sort of clonal disorders, start from stem cells. Okay, but during their evolution, because in CML some other genes get activated, so it becomes a CML with differentiating leukocytes, neutrophils, and other cells of the myeloid lineage, whereas a mutation in the stem cells that accumulates different kinds of mutations, additional mutations, will give rise to polycythemia vera and primary myelofibrosis. So there are different manifestations of myeloproliferative disorders, and there are different treatments for that. Okay, so we talked about the signaling starting from cell membrane and going through these intermediate pathways and mutation in these pathways. We talked about RAF mutations, talked about RAS mutations, we talked about growth factor receptor mutations. Ultimately, they all come to the nucleus. And the nucleus, all these signaling molecules have to activate transcription of genes which are required for cell cycling, such as MYC protein and cyclin. We'll talk more about that later on. Okay, so keeping in the same theme, there could be mutations in the genes within the nucleus. And yes, there are mutation activation. It does occur in nuclear transcription factors. So normally the nuclear transcription factors get active only when they get signal from upstream signals from RAS and other PI3 kinase and so on. Okay, but if there's a mutation in the transcription factor, okay, then the cell thinks that it needs to divide. Okay, the most commonly mutated in this group is MYC, activated in bucket lymphoma 814. It's amplified in breast, colon, and lung cancers, in neuroblastomas and small cell carcinoma of the lung. What does mutant MYC do? Mutant MYC upregulates cyclin-dependent kinases and inhibits their inhibitors, cyclin-dependent kinase inhibitors. We'll talk over that in a minute. Okay, it's not well appreciated, but it is becoming clear now. One of the major functions of MYC activation is to change the metabolic pathway of the cells, and we will talk about that altered metabolic pathway. Okay, so now there is no drug which can antagonize MYC, but there are drugs which are being targeted towards downstream targets of MYC activation, CDKs and CDKIs. Some are in development and some are in use. So ultimately the cell has to decide. All those signals have to come to the cell has to decide. So we should know something about what controls the cell cycle normally and what can go wrong in cancers. Okay. All right. So I'm sure all of you are familiar with the cell cycle. Cell cycle has 4 phases. Actually, the 5th one called G0. They're completely out of cycle, and then there is G1. And when a mitogenic signal comes, G1 has to go to S with DNA synthetic phase. At this place, there's a checkpoint. The checkpoint which controls that the DNA that has been duplicated by the growth factor, okay, does not have any errors. Okay, and it has to pass this filter, so that it goes to S phase, because once the cell goes to S phase, it can't stop. It has to divide after that. Okay, then it goes to G2 phase, and then in mitosis, okay, which is another checkpoint here, and then back into G1. So what happens normally? Cyclins, cyclin D, cyclin A, cyclin D. Cyclins are molecules which activate cyclin-dependent kinases. As the name indicates, these are kinases they can phosphorylate, okay, and they need to be signaled by activation of cyclins, which is shown here. Cyclin D, for example, activates cyclin-dependent kinase 4, cyclin kinase, and so on and so forth. Okay. So what do these cyclin-dependent kinases do? Well, what they do is this. They add a phosphate group to the RB protein, which we shall discuss shortly in more detail. And phosphorylation of RB protein removes this G1S block, and the cell can go into synthesis. So this is how signals coming from the cell surface have now reached the nucleus, where we have additional steps of control into cell division. So what happens in cancers? Well, in cancers, these things get dysregulated. Okay? For example, cyclin D is overexpressed in breast cancers, esophageal cancers, liver cancers, lymphomas, and myelomas. Cyclin-dependent kinases are amplified in melanomas, gliomas, and sarcomas. It's easy to understand. When there is such overexpression of cyclins, cyclin-dependent kinases, cell thinks it must divide. Cell thinks that it's getting growth signals from growth factors, receptors upstream of it. And so this is part of the cancer phenotype. Now, I know that, Psoriatic Newton said for every reaction there's an antagonistic to it. Okay. All our systems, physiological systems, are under very tight normal control. So it is not surprising that cyclins are regulated by cyclin-dependent kinase inhibitors. Okay. So CDK, and there are many inhibitors. Now, there are two classes of cyclin-dependent kinase inhibitors. One, which are called broad inhibitors. That's, for example, P21. P21 can inhibit all these cyclins, and this cyclins. Okay, cyclin B, cyclin A, and cyclin D. Then there are selective inhibitors. For example, P16. P16 is inhibiting only cyclin D. Nowhere else do you see P16. Okay? But anyway, that's their physiological function. Guess what happens in cancers? There's inactivation of these. There's inactivation of these inhibitors. When the inhibitors are inactivated, then the control over cyclins is lost. So there is somatic inactivation of P16 in majority of pancreatic cancers, glioblastomas, esophageal cancers, non-small cell carcinoma of the lung, and germline mutation P16 are responsible for familial melanomas, because these people are born with one mutant copy of P16. All they need is one more mutation, because there have to be double hits to tumor suppressor genes for their function to be lost. Okay, drugs are being developed, not yet fully developed. Drugs are being developed for overcoming the effect of these mutations in cyclin and cyclin-dependent kinases. Okay, so we spent quite a bit of time talking about sustained proliferative signaling, how it occurs physiologically, how mutations in various aspects of the pathway change the behavior of the cells, and how drugs are being developed. Some developed and some are being developed for treatment. Let's go now to what is called tumor suppressor genes which physiologically regulate cell growth. And tumor cells obviously figure out a way to evade this suppression. How do they do that? Let's just see. Okay, so one of the most important tumor suppressor genes is RB protein, encoded by the RB gene. And what does RB do? RB is a tumor suppressor gene. It monitors the G1S checkpoint. RB normally in quiescent cells does not allow the cell to move from G1 to S. And it does so because normally RB is hypophosphorylated. And hypophosphorylated sort of, almost like in a jaw-like fashion, entraps a transcription factor called E2F. This E2F gene is required, if it is released from here, to go and bind to specific sites in the DNA, which then allows the synthesis of cell cycle proteins like cyclin E, for example, as many S-phase genes. Okay, so in a resting cell, this is all quiescent. There's very little E2F coming out here. But what happens when a cell is given growth factors? Okay, cell is given growth factors, EGF, UEGF. It activates cyclin, okay, and cyclin-dependent kinases, which then phosphorylate the RB protein, which goes from hypophosphorylated stage to hyperphosphorylated stage. When it goes into hyperphosphorylated stage, it releases E2F. And the E2F then binds to its sites in the DNA, and S-phase gene transcription is activated. Okay? So we just went through this thing. Growth factor receptors. Growth factors lead to cyclin D expression and activation of the cyclin-dependent kinase 4 and 6, which in turn phosphorylate RB, which in turn releases E2F, and E2F can cause transcriptional activation. Okay. So mutations in RB gene make them in hyperphosphorylated stage. Okay. This control is lost, okay. And G1S block is lost. Okay. Now, dysregulation of G1S checkpoint is fundamental to cancer. So you might ask, if it is fundamental, does every cancer cell have mutations in RB? No, they don't. But every cell has a mutation in genes that control this important step. Okay, and let's see what they are. So G1S block can be overcome by 4 mechanisms. And one of these mechanisms will always be there in every cancer cell. Once again, we are saying not all cells lose RB by one mechanism, which means that the treatment of cancers in which RB function has been lost by different mechanisms have been treated differently. This again, we are moving towards sort of a type of personalized therapy. So what are the 4 possible ways? One is that some viruses such as HPV, which is oncogenic and causes or pharyngeal carcinomas and carcinoma of the cervix, they have the ability to bind to RB and sort of unlock the RB so that E2F is released. Mutations in cyclin D, cyclin D dependent kinases will have the same effect. Mutations in inhibitors of cyclin D kinases, CDKs, will have the same effect. So all these 4 things will have the same effect, and the effect will be this break is no longer there because the phosphorylation is no longer being controlled. So finding out in each cancer which of these pathways has been dysregulated, and then developing drugs appropriate for that will be a goal of personalized therapy. We come to P53, which is called the guardian of the genome. P53 is the most commonly mutated gene in human cancer. If you take all the genes, oncogenes, tumor suppressor genes, this is the one that is most commonly mutated and is vicious. P53 gene mutation is horrible. It's a horrible thing for anybody who has cancer. But let's just say why it is so. So this is a normal cell which has been exposed to ionizing radiations, carcinogens, mutagens, okay, which basically means the DNA has been damaged. Okay. Hypoxia can do the same thing. Then P53 controls DNA damage by 3 distinct mechanisms. 3 distinct mechanisms. Okay, what are these 3 distinct mechanisms? So when DNA damage occurs, the cell has sensors of DNA damage, and those sensors talk to P53, and P53 is activated and binds to DNA. Now, once bound to the DNA, 53, it can transcriptionally activate certain genes, and there are other effects of p53 which do not depend upon transcription. But what are those effects? An important one is transcriptional activation of P21, which is a cyclin-dependent kinase inhibitor. Remember, this was the one which was broad, affected most cyclin-dependent kinases. What does these do? Well, activation of this, their job is to cause G1 arrest, the cell cycle. Okay. So because they inhibit cyclin-dependent kinases, and so the cells are arrested in G1. Now, this is sort of to provide the cell rest, so that at the same time, DNA repair mechanisms have also been activated by 53. So P21 gives rest to the cell, arrest the cells and gives them period of rest, during which the DNA repair mechanisms attempt to repair the DNA damage of these cells. If it happens, cells are happy, things are back to normal. If this fails, then P53 says to BAX, which is a apoptosis-inducing gene, kill the cell. If repair fails, because the body cannot tolerate having cells with mutant DNA, because that's sort of the pathway towards cancer. So there are two effects here. The third one is to a different pathway. Okay? The DNA damage leads P53 to put the cells in a quiescent stage by inhibiting translation of growth promoting genes. And the cell is not transformed, not completely normal either, but is quiescent. It won't divide. Okay. And it can also, P53 can induce apoptosis of cells by a mechanism slightly different from the one shown there. So these are the three ways that P53 protects the genome against DNA damage. So you can imagine, loss of P53 is going to be drastic. So what happens with loss of P53? DNA damage occurs, it remains unrepaired, there's no cell cycle arrest, there's no repair of DNA. Mutant cells with damaged DNA can acquire additional mutations and evolve into a clonal but heterogeneous tumor. How often does this occur? It's very often. And typically, cancers in which P53 is mutated are very difficult to treat, because most of the chemotherapeutic drugs function by inducing apoptosis. And germline mutations in P53 cause the Lee-Fraumeni syndrome. And so just to step back for a moment, I think I mentioned to you that for tumor suppressor gene action to be abrogated, both copies have to be mutated or deleted. It's called the Knudsen's two-hit hypothesis. Those who are born with one mutant copy, okay, they need now only one more mutation, and which is there. So getting two mutations is less likely than getting one mutation. So if patients who are born with germline mutations of P53, they are prone to get a second hit to their P53. And at a very young age, these individuals come up with multiple cancers, lymphomas, carcinomas, all organs, because P53 functions in every cell of the body. Okay, so cells can grow by promoting growth factor signaling, making the cells not sensitive to growth inhibition. Another way they can do it is by evading apoptosis. The cells don't die, and even if they don't divide very, very rapidly, they'll accumulate. And the classical example of this is upregulation of the BCL2 gene. BCL2 is a family of genes, some of which are pro-apoptotic, some of which are anti-apoptotic. Okay. BCL2 is an anti-apoptotic gene. And if its expression is upregulated, as occurs in follicular lymphomas, the 1814 translocation. then the cells don't die in response to normal death signals, okay, which again contributes towards malignancy. And there are other examples which I won't go into. Okay, coming back to this diagram of the hallmarks and enabling features of cancer, we now go to one of the more recently understood hallmarks of cancer, that is deregulating cellular energetics. Okay, here we go. So, reprogramming of energy metabolism, a new hallmark of cancer. New means last 8-9 years, okay, as opposed to 15-16 years. So one example is here. In normal cells, glucose metabolism occurs by oxidative phosphorylation in the mitochondria. We all know that, under aerobic conditions. And when there's hypoxia, there is anaerobic glycolysis, which sort of goes to the pyruvate pathway rather than the Krebs cycle pathway completely. Okay. So when normal oxygen is there, this is occurring, oxidative phosphorylation. And when there is the paucity of oxygen, the cell triggers for glycolysis. Now, interestingly, tumors shift glucose metabolism from aerobic mitochondrial to aerobic glycolysis, even when there is enough oxygen. And people often mistakenly call this anaerobic glycolysis. No, this is glycolysis occurring in the presence of sufficient oxygen. So that's why it's called aerobic glycolysis. Aerobic glycolysis is pathological. Anaerobic glycolysis is physiological. So even when there's enough oxygen, tumors shift their metabolism of glucose from mitochondrial to aerobic glycolysis. This phenomenon was observed way back, and Otto Warburg, a German chemist, got the Nobel Prize for discovery of this aerobic glycolysis. But its full impact didn't get understood until a few years ago. First of all, a clinical application of it. Aerobic glycolysis produces much less ATP per glucose molecule when compared to mitochondrial phosphorylation. So mitochondrial phosphorylation that occurs normally in cells under aerobic conditions creates about one oxygen, probably 38 ATPs by aerobic glycolysis. By oxidative phosphorylation. But when aerobic glycolysis occurs in tumor cells, when tumor cells switch to aerobic glycolysis, even in the presence of oxygen, the number of ATP molecules per glucose molecule is much less. 2 ATP molecules as opposed to, or 30 ATP molecules from per glucose. What does this do? This leads to increased hunger of cells for glucose. So they increase the uptake of glucose to meet the energy needs. And we have exploited that to do PET scanning. Glucose hunger can be seen by PET scanning, in which an enhanced uptake of radio-labeled glucose can be seen to enter the tumor. This is basically a imaging reflection of aerobic glycolysis occurring in this tumor. Why does it occur? Why does it occur? Why does it why do tumor cells make less ATP? Okay. They make less ATP because tumor cells must not only make new DNA, but they must also make all other cellular components such as amino acids and lipids are needed. As cells are dividing, they need not just glucose, not just DNA, but all other endoplasmic reticulum, mitochondria, and so on and so forth. And in aerobic glycolysis, the glucose breakdown stops at pyruvate, okay, and this allows the remaining carbon atoms of the glucose to be shunted for other anabolic activities and synthesis of organelles in dividing cells. You might say, what happens in an embryo, which is rapid dividing? Exactly the same thing happens in an embryo. There's aerobic glycolysis for exactly the same reason. What's the difference? In embryogenesis, it is controlled. In carcinogenesis, it's uncontrolled. . Tumor cells must develop blood vessels, must sustain angiogenesis. Otherwise, they can't grow beyond size about 2 centimeters. Okay. So how do tumor cells sustain angiogenesis? Well, that turned out to be the topic for which this year's Nobel Prize was granted. Normally, angiogenesis and anti-angiogenesis are balanced. So, for example, in a healing wound, one sees granulation tissue. There's angiogenesis going on there. But when the wound heals, those vessels shrink, everything is replaced by collagen, and you see a scar. So there's a system for angiogenesis and a system for anti-angiogenesis. And physiologically, they're balanced. In tumors, this switching of angiogenesis and angiogenesis becomes disordered. And that balance tilts towards angiogenic and not anti-angiogenic. Okay. So that there is continued angiogenesis. Obviously, the tumor needs it. And so tumors have to figure out a mechanism by which to make sure that they keep on getting oxygen and blood supply. So there are several mechanisms for this. One, which was the topic of this year's Nobel Prize, is this. So how does this occur? Hypoxia upregulates the transcription of a transcription factor called hypoxia-induced factor alpha. It goes up. Hypoxia-induced factor alpha has the capability of transcriptionally activating vascular endothelial growth factor, VEGF, which is an angiogenic molecule, which will make new capillaries. Okay. Normally, this process is controlled. How is it controlled? A gene which is called VHL downregulates HIF1 alpha. So this downregulates HIF1 alpha, goes down, VEGF goes down, the process remains controlled. Okay. So you have guessed it. Mutations in VHL, which give rise to the VHL syndrome, von Hippel-Lindau syndrome, where there are, renal cancers and the medulloblastoma in the brain and several other things. Because patients with VHL syndrome are born with one mutant copy of VHL. Then they lose the second one, and this whole system goes awry. There is sustained VEGF and sustained HIF1 alpha, and the tumor thinks that there's not enough oxygen and keeps on creating more and more capillaries for blood vessels, blood supply. Now, VHL is not the only gene that controls angiogenesis. Mutation in RAS and TP53, which are very common, also promote angiogenesis by slightly different pathways. What's important is this, that angiogenesis is so central to tumor growth and survival that we now have an armamentarium of anti-VEGF drugs, which are given in a variety of cancers. Activation of invasion metastases is also an important hallmark of cancer. And we all know this sort of diagram in which there's a journey of cancer cells starting from the primary tumor. Notice what's labeled here, metastatic subclone. This population is heterogeneous. Not every cell in this has the capacity to metastasize. So this metastatic subclone, which has developed the properties to adhere to the base membrane, okay, how does it happen? It must loosen the intercell attachments. And what keeps the cells attached to each other? EK inhibitor. So loosening of tumor cells so that they can metastasize is related to loss of EK glue. How can it be lost? Mutation in EK genes can do that. Okay, and the EK gene mutations are seen often in gastric carcinomas, familial loss of EK. EK signals through beta-catenin. So a mutation in beta-catenin has the same effect as mutation in EK. And this occurs in many cancers, colon cancer, and so on. There's also a mutation of a couple of genes with this interesting names called snail and twist. they are mutationally activated. Now, what do these genes do? They cause epithelial to mesenchymal transition. They change the properties of these epithelial cells to mesenchymal cells. What does it do? It gives them more mobility. It gives them the microfilaments that are required for locomotion. Epithelial cells are not moving cells. Mesenchymal cells are moving cells. Fibroblasts can move, for example. Okay? So there's epithelial to mesenchymal transition, which is seen most clearly in breast cancers by the activation of snail and twist. So these genetic abnormalities listed here allow the first step of metastasis here. Then the cells have to, go through the stroma. They have to secrete MMPs, which are proteinases. And that releases growth factor from ECM and motility factor from the stroma. So not only is the stroma being dissolved, other things are happening. Growth factors are coming out of ECM and from the stroma. And then there's vascular dissemination and homing. Okay. The only thing I want to mention here is this, which you probably all know, that metastasis is not always to geographically determined, that is, breast cancer to lymph nodes. Yes, we know that. Okay, but we also know that lung cancer goes to adrenal glands, and we also know that breast cancer goes to bones. Okay. So metastases can also be explained on the geography of the tumor. Tumor cells apparently have properties where under the influence of chemokines, they can go to selective sites. And that might be an area people might think about working to prevent metastases, but it is still not fully understood. Okay, we now come to the two enabling hallmarks, genomic instability and tumor-promoting inflammation. Genomic instability occurs when there are mutations that create defects in DNA repair. If mutations affect DNA repair mechanisms, what does it do? Those genes itself don't do anything directly. They allow unrepaired mutations in genes that control hallmarks of metastasis. All those genes we talked about. Okay, mutations can tend to sort of dysregulate their function, but that is normally prevented by the DNA repair mechanism that we have. Absence of this, patients born with defects in DNA repair genes are at greatly increased risk of cancer because they can't repair those mutations. And a very good example of that is hereditary non-polyposis cancer syndrome, HNPCC, which is caused by mutations in genes involved in DNA mismatch repair. In xeroderma pigmentosum, the mutations affect nucleotide excision repair. In familial breast cancers, mutations are in BRCA1, BRCA2, which control homologous recombination pathway. There are many pathways of DNA repair, and these exemplify three pathways in DNA repair, and the genes that are involved in it, which when mutated, give rise to cancers. So this itself is not a hallmark, but allows other hallmarks by not being able to repair the mutations in those genes. Tumor-promoting inflammation. This is a section from lung cancer. And I think many of you may have seen, in those of you are residents and practicing pathologists, often there is a lot of inflammatory cells. This is an area of lung cancer where there's no necrosis, but there are a lot of neutrophils. And it was thought for a long time the infiltration of leukocytes in the tumor cells is a host response to tumors. It's an inflammatory response, an immune response to tumor. Paradoxically, it seems that inflammatory cells may also promote tumor growth. So this is an enabling hallmark by secretion of growth factors such as EGF and pro-angiogenic molecules such as VEGF, secretion of MMPs that digest the stroma, release of free radicals which can inflict additional DNA damage. So, question comes, if this is something which is enabling hallmark, can we control it? Well, there is some evidence right now. Might inflammation control be therapeutic target? Actually, many of you might know that COX-2 inhibitors have been used, particularly for preventing recurrence of colon cancer. And particularly in patients who have polyposis, to slow down or prevent that transformation into invasive cancer. How do they do this thing? Obviously, COX-2 inhibitors, inhibit prostaglandin synthesis. Prostaglandins do all sorts of things, in the stroma of the tumor. So whereas these are not yet established as very common ways of treating tumors, they are being actively investigated for that. We come to avoiding immune destruction. Okay, this is interesting. This is also something for which there was a Nobel Prize a couple of years ago. For many, many, many years, people thought the reason why the immune response cannot get rid of tumors is, number one, tumors don't have any new antigens, so there's nothing for the immune response to see. The other was, no. They have new antigens, but they can't but they can't make cytotoxic T cells. Okay, they suppress mechanisms involved in developing cytotoxic T cells, specific for the tumor, so there's nothing happens. The third one was, tumors have antigens. They can evoke the formation of CD8 positive cytotoxic T cells, CD8 or other T cells, but something prevents these T cells from killing the tumor. Okay, and that turned out to be correct. That turned out to be correct, leading to one of the most spectacular advances in treatment of human cancer that has occurred in the last 10 years. So most cancers do evoke CDL-CTL response, but cannot kill tumor cells because tumors hijack normal checkpoints of T cell activation to avoid being killed. So here is a CD8 T cell. Here is a tumor cell. Here is an antigen of the tumor, which the T cell receptor of the CD8 T cells can recognize. But guess what? What else is happening? There's a receptor on T cells called PD-1. It has a ligand called PD-1 ligand. This receptor turns off T cell activity. So if the tumor expresses PD-1 ligand, okay, the PD-1 receptor on T cells recognizes it and shuts off this signal. So the tumor cell cannot be killed. Okay, how can this be overcome? Well, by giving anti-PD-1 antibody so that it prevents this interaction, or antibody to PD-1 ligand. These are currently being used in immunotherapy. They've been phenomenally successful in melanoma. They've been phenomenally successful in melanomas and non-small cell lung cancers. Okay. And their use is increasing. Their use is increasing. Because once we have come to know that tumors have antigens, in principle, this should work. Okay, coming towards the end, what are some inadequacies of current cancer treatment? Morphology is the dominant current tool for cancer treatment, which does not accurately predict the fundamental genetic changes in a given cancer. Okay. Morphologically identical cancers can be genetically distinct, and conversely, morphologically distinct cancers may share a common genetic effect, just as I gave you the example of BRAF mutation. Relatively few specific mutations have been successfully targeted by treatment. In breast cancer, Herceptin, chronic myeloid leukemia, some lung cancers. The question then comes, does cancer genomics, the study of entire genome of the genetic architecture of the cancer, offer hope? And two approaches have been applied. One is expression profiling of tumors by DNA microanalysis. Basically, this is a way of interrogating thousands of mRNAs at the same time. Okay. This is basically a simultaneous measurement of expression levels of thousands of genes. You can then compare them with the normal, but more importantly, you can do such an analysis. For example, it was done first on large B-cell lymphomas, and the expression array analysis revealed that actually what appears to be a homogeneous group of large B-cell lymphomas is actually 3 subgroups. This was the first application of interest done at Stanford. Expression profiling has not become very common because technically it is very tedious. And so it can't be sort of done in a routine way. Only very specialized labs can do this thing. The next is high throughput next generation sequencing of tumor DNA. And this can reveal the mutation profile of the individual cancers and may lead to, identification of genes, which might be unique targets. If you look at the genome of every cancer, you might find such rare gene mutation that might be an appropriate target for that particular tumor. So, but quickly, expression, RNA expression analysis, DNA microarray analysis, just an example of this one. Okay, top one, expression profiling. So look at the morphology. These are three different tumors. They look pretty much the same. Same types of cells, same sort of starry type appearance, etc. Okay. Is it a bucket lymphoma? Is it an atypical bucket lymphoma? Is it a diffuse large lymphoma with P14 translocation? That was the differential diagnosis offered by several experts. So it's actually done. So morphologically identical tumors are genetically distinct. And when DNA microanalysis was done, okay, and I won't go through this complicated chart, different patterns emerged in these tumors. Some were bucket-like, some were large B-cell-like, okay, and some were of a different type. So clearly, these 3 morphologically identical tumors didn't have the same genetic changes because the treatment for bucket type versus diffuse large B-cell type is quite different. Expression analysis, microanalysis has found its use in expression profiling of breast cancers. Okay? And this is an interesting example of something which was sort of done initially in a research base, that is taking 28 genes and 26 breast cancers and looking for that expression. Okay? 4 groups stood out. Just compare the number of reds and greens. And these 4 groups corresponded to the 4 well-known subsets of breast cancers: luminal A carcinoma, lobular carcinoma, HER2 positive carcinoma, and triple negative carcinoma. We no longer have to do this thing in the lab because it's already established, and these can be separated by immunohistochemistry. Okay. And you can say immunohistochemistry is also a way of expression analysis at the protein level. It's a proteomic level rather than at the genomic level. Finally, cancer genomics. We now have the capacity to sequence the entire tumor genome in a relatively short period of time at relatively low cost. This could create a mutational profile for each individual cancer. And that may allow identification of targets unique to the individual. We previously discussed there are 2 types of mutation in tumors: driver mutations and passenger mutations. Driver mutations can be targeted because they are driving all the main properties of the tumor. Passenger mutations, not. And so sometimes, the genomic people have to distinguish when they look at the pattern of mutations by doing next generation sequencing, which is driver, which is passenger. There are algorithms for that. Just a caution, although driver mutations are defined as recurrent and occurring in a large percent of patients, there are driver mutations which are present only in a small percent of tumors. For example, the AML ALK fusion gene is present in many T-cell lymphomas, but only 4% of non-small cell carcinomas. Now imagine, how would you have imagined that a mutation that drives T-cell lymphoma will also drive lung cancer? Nobody would have thought about it. Okay. But having discovered that, these 4% of the patients now can get a different kind of therapy compared to everybody else. And that therapy given to others would be useless and harmful. to these patients, it will be useful. So this is really a very nice example of a targeted therapy coming out of genomics. So what should we be doing? I'll tell you what we are doing. We have a 1,200 gene panel on a CLIA platform. Every cancer that is operated upon at University of Chicago, and every sort of FNA, and every cytology smear which has at least 5,000 cells, is interrogated for mutations in these 1,200 genes. How did we come to these 1,200 genes? There are 3 buckets in it. The top bucket has 150 genes which are actionable mutations, which are known to be actionable, okay, in various tumors. Okay. So we don't actually have a tumor-specific panel. We have a panel which has all actionable genes, and then depending on the organ from which the cancer came, we can identify which are the actionable mutations in that by using informatics. Next is a bucket of genes in clinical trials. The idea is this, that once these clinical trials are over and they show benefit, we can very quickly start targeting them without having to do sequencing again, because we didn't do the first sequencing in a CLIA platform. You can't make any clinical decisions on a test that is not run on a CLIA platform. And so we said it's likely that some of these genes in the clinical trials will become useful for targets, so we'll do it in a CLIA environment. So once one of these candidate genes are identified, they move into this category, and we have tell the clinicians you can treat it now. And the next bucket of genes are in discovery phase. And our expectation from these will emerge these, and from these will emerge these. And from that will emerge personalized therapy for the individual. Over time, genes move up in the buckets. Since all data are obtained in CLIA platform, it can be used for treatment. The most exciting thing, the most really exciting thing, is this National Cancer Institute Genome Data Common. So what the National Cancer Institute has done is they selected University of Chicago as the home base for this, although there are branches of this in many places in the country. All the sequencing data available anywhere, okay, is put into this National Cancer Institute Genome Data Common. Imagine, for example, it has probably sequencing data from 1,000 breast cancers. No single institution can do that. Okay, so why is it important to have sequencing data on 1,000 breast cancers or 500 lung cancers? It's useful because when we are doing sequencing of tumors at the University of Chicago of a breast cancer, and we find a variant that looks unusual, variant by definition is something which is not normally there. We ask ourselves, is this variant clinically significant for this patient? Okay, we don't know. Nobody has published anything like this. What do? We go to the National Cancer Institute Genome Data Common and say, compare this to all the sequencing data of 1,000 breast cancer patients. And sometimes it does happen, somebody else has also seen that. Maybe we find 10 cases in the genome database which have exactly the same mutation. Okay. Now we begin to say, this can't be just any passenger mutation. The likelihood that by chance it occurred in 50 other breast cancers is very small. Then we can go back and look at the clinical features of those patients and maybe define a subgroup, and then maybe develop a drug for a genetic morphologic subgroup. This is the goal of personalized medicine. This is where we are moving. It's exciting. It's very exciting time, and particularly for those who understand disease mechanism, pathologists and other clinicians as well. Okay, I think I've taken about 10 minutes longer than I should have. I apologize for that. And I won't talk about epigenetic modifications. That's a whole subject. A gene expression is changed not just by mutations but also by epigenetic phase. So cancer genomics, the goal is targeting driver gene responsible for hallmarks of cancer. And interestingly, drugs are being developed for almost all of them. Cyclin-dependent kinase inhibitors, telomerase inhibitors, anti-inflammatory drugs, PARP inhibitors, VEGF signaling inhibitors, treatments that mimic the effect of pro-apoptotic genes, targeting aerobic glycolysis, targeting growth factor receptors, targeting immune blocking molecules. Common driver mutations shared by heterogenetically distinct tumors. Paradigm shift. Classification of cancer according to therapeutic targets rather than cell of origin. Are these tumors BRAF-formers? I already answered this question. Yes, they are BRAF-formers in one sense, but in cancer, everything is not that simple. Not all of them can be treated with BRAF inhibitors, but there are other therapies being developed for that. Finally, I'd like to acknowledge the support of Dr. Anil Prasad, president of ATMA, who came up with this idea in consultation with Rifat Manan, who has been involved in PathCast, and Emilio Madrigal, Rifat and Emilio have made this possible. And Dr. Anil Prasad has been a champion of this, and I'm thankful to all of these. Thank you. Very interesting. Very interesting. Okay. I actually do not know those specific proteins. I completely understand why the question is being asked, because I actually alluded to the fact that one could target that. Okay. Unfortunately, that's a very specialized area in which I don't have sufficient expertise to answer a question, but I think it can be found out. Oh yes, there's absolutely no doubt. So as mentioned, that in the United States, National Cancer Institute has spent money and resources and recruited people from people at our institution, other institutions, to do the informatics that's required. Let me just tell you a challenge in doing this thing. The challenge is this. So if you take data on breast cancer from UK, from Netherlands, from Australia, from India, from Canada, from Brazil, okay, most of those have been collected using informatic pipelines, which they developed themselves. Okay, you put them all together, you can't read them, because unless you know how to read the Brazilian data and the Australian data and the UK data, it's useless. So what the informaticians have had to do is to put this data in a form that can be queried along a single informatic pipeline. And that is a very expensive and very tedious process, but it is being done. It's being done. Now, should this be done for the entire world? Absolutely. Absolutely. It should be done. I'll give you an example. There's someone at our institution who is studying breast cancer in Nigerian women and breast cancer in African American women in the US, and trying to find out racial and ethnic differences which will impact on their treatment. So if we had a sort of an international database like this, we could also sort of think of developing therapies which may be more appropriate. I mean, for example, oral cancer is very common in India because of betel chewing and smoking and so on and so forth. Oral cancer is less common in the US. Should they be treated the same way? Most likely not. Most likely the cancers that arise in the US and in India are driven by different carcinogens. Okay. We know that, for example, exposure to aflatoxin causes very distinctive mutations in P53. In fact, by finding that mutation in a tumor, one can almost predict that this person must have been exposed to aflatoxin or hepatitis B. So there are many benefits of doing such large-scale analysis, but again, it's highly expensive, and somebody has to spend the money.
Facebook page, as well as subscribe to our Pathcast YouTube channel. And we will be hosting more lectures in collaboration with IPNA, so stay tuned for them as well. And thank you again, Professor Kumar. It was a pleasure having you.
Talk to you again sometime.
Thank you.
Okay.